Hey, so you're at home, and this is the kind of the mandatory assignment for today. So I want to talk to you about the central dogma of biology that we started yesterday. Remember, that's the DNA to RNA to protein. And we're also going to look about the history and structure of DNA. So going into a little bit of the history, it was actually an international race to see who could discover the structure of DNA first. Really, the main focus was in the United States and in England. But eventually in 1953, specifically April 25th, 1953, two scientists by the name of James Watson and Francis Crick, which we saw their very famous photo yesterday in class, ended up creating a model that was correct for the structure of DNA. And what their model suggested was two chains wrapped around one another like a double helix or a spiral staircase. So double helix meaning two, double meaning two, and then helix meaning spiraled. So again, they were in like a lab conducting research. They used evidence from other scientists to help build their model. And two of the scientists that they received evidence from was Rosalind Franklin and Maurice Wilkins. Rosalind Franklin, her photo is in my classroom, if you ever have looked at that. But she specialized in X-ray crystallography, so what she would do was take the crystallized form of DNA and take photos of it, specifically X-rays of the DNA. And in that DNA fact sheet that you used yesterday in your model building, there's a photo in that fact sheet, and it's specifically called Photo 51. So this is a very famous photo that Watson and Crick used in order to determine the structure of DNA. So several years went by, about 9 years, and Watson, Crick, and Wilkins ended up winning the Nobel Prize in 1962 for the discovery of the structure of DNA. But unfortunately, Rosalind had died before that. She died of cancer, so she is not given credit for the Nobel Prize. So she died of cancer because when she was taking all those X-rays, she wasn't wearing any protection. Like, if you go and have an X-ray, maybe like you wear lead vest or something like that. She wasn't protected by anything, so all that exposure from the radiation caused her to have cancer. So some other scientists before Watson and Crick that also helped determine DNA is our genetic source were Frederick Griffith, Oswald Avery, and then Hershey and Chase. So a couple of these you read about in your reading guide. Frederick Griffith did the experiment with the mice and the RNS strains, and he discovered a transforming principle. He thought it was DNA but couldn't verify that. A couple years later, Oswald Avery repeated Griffith's experiment, and he did confirm that DNA was that transforming principle, not a protein. Then eventually Hershey and Chase used viruses, specifically phages. Remember, a phage is a virus that attacks bacteria. And they were able to confirm as well that DNA is genetic material, not protein. All right, speaking of DNA, we know that DNA stores genetic information that can be passed on from parent to offspring. That's what heritable means. And here we see that central dogma of biology, from DNA to RNA to protein. I just added in there amino acid as a reminder that amino acids eventually form proteins. Remember, the amino acid is the monomer of a protein. And as you saw yesterday, when you all figured out that we have the sugar, the phosphate, and the base, as far as that pattern, I think the light bulb really clicked for a lot of you all. And so remember, with any nucleic acid like DNA, the subunit for that, or the monomer, is the nucleotide. And remember, the nucleotide has 3 parts: a 5-carbon sugar, specifically for DNA, that's deoxyribose, a phosphate group, and one of 4 nitrogen bases. As you saw in the models yesterday, we had our 4 color-coded pieces that represented our bases in DNA. So we had, I think, blue, red, gray, and green for those pieces. And it was important, too, when I was checking some models, that I would look at these colors and how they paired. As far as the strands or the sides of our DNA, we call that the backbone. So we talked about this yellow piece yesterday, the covalent bond. So you can see in this model here, they alternate phosphate, sugar, phosphate, sugar, phosphate, sugar. So that makes up the chain or the backbone of DNA. And then we can see, as a lot of you figured out yesterday, the base specifically comes off of that 5-carbon sugar of deoxyribose. As far as looking at the structure of DNA, it is a double or spiral helix. Double means, again, 2 strands. So in this picture, I can see 2 strands kind of wrapped around one another to form that spiral shape. One other thing that was in your fact sheet was Chargaff's base pairing rules. So Chargaff, I think, was a French scientist who looked at DNA from different species, and what he concluded was that the percentages for adenine and thymine were always similar in the same species, and then the percentages for guanine and cytosine were also similar in the same species. So we call these Chargaff's base pairing rules. And if you remember that white piece from yesterday, the white piece is that hydrogen bond that bonds our 2 bases together. So this is what I was trying to get at yesterday about your sequencing or the order of the bases. If I just look at one chain of my DNA, the sequence of my bases here does not have to be repetitive. If you see GCTA, it doesn't go again GCTA. The sequence here does not matter. So the order of the bases contains the message for controlling cell processes. So this is where that genetic code is stored. So it's ultimately the sequence of these bases, how those A, T, Gs, and Cs are arranged, that leads us to RNA, that leads us to protein. So I like this image here. We'll talk about this when we talk about protein on Friday, but the 3-letter words, the genetic code, are the same in every creature. CGA means arginine, and GCG means alanine in both bats, beetles, beech trees, bacteria. They even mean the same in the archaebacteria living at boiling temperatures in sulfurous springs thousands of feet beneath the surface of the Atlantic Ocean, or in those microscopic capsules of deviousness called viruses. All life is one, seaweed is your distant cousin, and anthrax one of your advanced relatives. The unity of life is an imperial fact. So notice it has kind of our tree of life here that stems from DNA. So this is a source of evolution, or source of evidence for evolution, that all living things share are those 4 bases of A, T, G, and C. So one other thing that Watson and Crick developed from their model was, besides just the structure, is that how is that genetic code passed from parent to offspring, or how is it copied? And that is called replication. So the purpose of replication is to make copies of DNA, and those copies are identical to one another, and that process occurs during cell division. So during division, each cell made needs copies of DNA as the parent, and those copies are going to be exact. So each strand of the double helix serves as a template or a pattern to create new strands. And so this method of replication is called semi-conservative because it's conserving half of the original strand and then producing a new strand. So this is what Watson and Crick also developed. There we go. So there are a lot more steps to the process of replication besides this one I have up on the screen, but these are kind of the main steps. So first of all, we want to access those bases that are in the middle of DNA. So we actually need DNA to unzip or unwind, and the enzyme of DNA helicase comes along and kind of breaks those hydrogen bonds. Remember, the hydrogen bond is a weak bond, so it's easily broken or easily reformed. And so the enzyme DNA helicase is going to come along and break that bond. So each side of the chain is now accessible. The new bases are added in both directions along the original strands. So think of it as a divided highway. If you're driving like on the interstate, you have some cars going north and then some cars going south. So bases are added in both directions to each of the original strands, and an enzyme called DNA polymerase helps with adding those bases. And then the process continues until there are 2 new complementary strands produced. So within each strand, half of that is original, and half of that is a newly replicated daughter strand. So what that kind of looks like is I have my original copy up here of my DNA. So it says parent DNA molecule, 2 complementary strands of base paired nucleotides. So I can see Gs and Cs and Ts and As. And then we're going to unzip our DNA here. It's called a replication fork. And parental strands unwind and separate at several points along the DNA molecule, forming replication forks. So around here, you can see it's no longer spiraled. There's a big gap here. Number 3, each parental strand provides a template that attracts and binds complementary bases A with T and G with C. So you can see this kind of darker blue on both sides represents the original parent strand, and the kind of lighter blue are the new daughter strands. And that process will continue until number 4, the sugar phosphate backbone of daughter strands are closed, and each new DNA molecule consists of one parental strand, kind of the darker blue, and one new daughter strand, that lighter blue, as a result of semi-conservative replication. So these are the notes that you're going to be going over today, and we'll review them tomorrow. We'll review them on Thursday, but make sure that you go through these notes and be prepared on Thursday, and I'll see you Thursday. Bye.